This one should be quite cool. It's a update on a topic that is near and dear to many of y'all's hearts, right? Is on this animal here. It is the thylacine, folks. What is a thylacine? Why do we care about the thylacine? Well, that's a great question. It is an animal from Australia, and it was the only marsupial apex predator. The last known thylacine died in captivity in 1936 because what? People killed them all. Why did they kill them all? Because they were a bother to farmers, sheep, whatnot. There are the occasional suspicions that someone has come across a thylacine in the wild. There is nothing to suggest that that has actually happened. But there is this program here from Colossal Laboratories, which I will link y'all if you would like to check out. This company is trying to bring back the thylacine. They're trying to bring them back to the forefront by actual cloning. And so we're going to go into a little bit of an update of what they want to do and how they're planning on doing this. And there's actually the update I think is really cool because now they're getting closer and closer of de-extinctifying this animal. So the last time we talked about this, y'all, was maybe 2 years ago. And it was from the University of Melbourne. There was a paper that came out from the PASC lab that was trying to cause there to be de-extinction of this animal. And they partnered with this group Colossal to be able to do that. So the new advancements is that they've actually made a much better genome. So with a bunch of sequencing, they got it to be 99.9% accurate. So sequencing a bunch of different animals and getting these sequences put together in a way that you can be like, all right, we have the correct sequence. If we put this into the embryo, it will be a genetic copy of the thylacine. If an animal has gone extinct, is it possible that nature has changed in its absence? And it is an irresponsible endeavor to bring them back from extinction. Absolutely, Professor Falcon. They're very difficult to address. For example, there is the argument that we should bring it back because we are the ones that drove it to extinction. In which case, we should also bring back the animal like the dodo and the passenger pigeon and the mammoth because we drove it to extinction. But then there's the counter argument, like you're pointing out. All right, sure, if we drove it to extinction like 10 years ago and we bring it back, like maybe the Earth hasn't changed that much in 10 years, but if it's 100 years, is it going to live? Is it adapted for living in this environment? Is there going to be something negative by bringing it back? Not done these things in the first place. So, Falcon, that's a great question, right? Let me give you an example, Falcon, where it's worked. There is the black-footed ferret. The black-footed ferret was hunted to near extinction in the United States to the point where there were 20 left in the wild. Only 20, Streaming Falcon. And researchers were like, all right, do we let them all die or do we bring them in and breed them and save them? So they brought in all 28 of these black-footed ferrets and started breeding them. They bred them together and started a whole breeding program, try to get these animals to take off in terms of that breeding program. And now they're releasing them into the wild, Falcon, and their populations are going up. But one thing that happened was if you only have, there were only 28 total, and I think half of them were female, and only half of those were actually being able to be bred in captivity. They're not very genetically different. They're very inbred. And so what ended up happening was researchers cloned a black-footed ferret from a specimen that was collected in the 30s. So this ferret genetically had not been alive for 80 years. And they cloned her and then introduced her into the population so that if she bred, she would introduce new genetic diversity to the population. And so is that something, Professor Falcon? They're still alive. If we don't intervene, they're going to be inbred and there's going to be just like destruction of their species. Is that something that we should go do? Is that worth our time, effort, and energy? I'm not saying one way or another. I'm just throwing that out there as a hypothesis. If we also talked about how much animals have been made extinct, how they change the environment, like the mammoth, compacting the tundra, we killed it. Yep, and that changed the habitat. And Jay, that's why we're trying to bring back the woolly mammoth. So Falcon, I don't know if you've been here when we talked about this, but there's a lab at Harvard who's trying to bring back the woolly mammoth. And they're not actually trying to clone the woolly mammoth as like 100% clone. They're trying to take an Asian elephant's genome, change 50 genes, and after those genetic changes are done, implant that embryo into an Asian elephant, give the Asian elephant to give it birth, and then you have an Asian elephant that's very hairy and maybe behaves like a woolly mammoth. Like that's so all that the guy wants, only the guy what the guy wants isn't to bring back the woolly mammoth, it's to bring back an animal that will behave like the woolly mammoth to stomp down the tundra. Yes, it's a huge area, Chris. The other thing that, Chris, how do we know that they're going to know to do that? So, Chris, I can maybe identify the genes that'll make the animal hairy. I can maybe identify the genes that'll increase its fat storage. There is no way in heck I'm going to identify the genes that are going to control the behavior of the animal to stomp down the tundra and the permafrost. No way that I'm going to predict that. And the issue, Jay, is if you bring those woolly mammoth-like creatures, like if you make a bunch of them, they've calculated they'll need to make 40 to get a breeding population that's not going to be inbred. Bringing all those in the world at the same time, like it's a difficult thing. The cool thing, though, that they're trying to identify, though, that I think is really interesting is an ex vivo uterus. So instead of growing the clone in an actual Asian elephant, they're wanting to just grow the clone in like a bag outside of the actual animal to see it develop. And that could be really, really cool. Climate change has altered the tundra far more than the extinction. And Neil, the argument, though, is that if you don't stomp down the permafrost, you're going to release all this methane and CO2 buildup that's stored in the tundra, you're going to cause even more climate change. And yeah, so the argument here, Jay, is that if you don't bring back the woolly mammoth, the environment will continue to change because the animal's gone. And it's like there's a window of time to correct it by bringing back the animal. Is that a sufficient enough reason to do this? Jay, I don't know. I don't think we'll ever know. But it's still just a really interesting philosophical question, if you will. But Void, listen, so like I guess the argument is, if you can get an ex vivo uterus to work, maybe you can apply that to other organisms as well. Not necessarily to the mammoth. Like maybe, like in principle, maybe they would start the ex vivo uterus experiments with something else, right? And then if you identify it, are going to be like, right? Like I can't predict if this is going to work or not, or if they're just going to do something different, right? Because we've done this with invasive species. There's a bunch of bunny rabbits out here. Let's give the predator to the bunny rabbits, and then they'll kill the bunny rabbits, and that's great. And then the predators are like, oh no, I don't need the bunny rabbits. I'm going to need whatever is actually endemic here. And then everything backfires. So it's not ideal. But again, at least in like this kind of upbeat, is that they've identified they've completed the genome 99.9%. They've also edited 30 markers of genes. So like here's the catch with these animals, Jay, the thylacine, is that they need a surrogate. They need a surrogate for this to work. And the surrogate is this animal here, which let me grab the... So yes, it looks very strange, doesn't it? This is the surrogate host. So what they actually did was they took cells from this animal and edited 300 different genetic components to make it as close as possible to the thylacine, which I think is really, really cool. They do point out issues that there's going to be for like the breeding capacity of these animals once they're in captive populations, once they're released in the wild, and more too. Let's chat about de-extincting this animal. Again, this video is a little bit on the older side, but what's really cool is the new updates that have been identified are really, really cool because you've expanded the knowledge of the genome. So the cloning that we're going to be doing is closer to what the actual animal is going to be like. So they are doing it in the responsible manner. There's enough positive aspects that we should at least try. I think, Jay, the nice thing about this technology is that all the advancements that they've made in this cloning process is neat because it'll help other animals that are on this path, like the Tasmanian devils, especially with the viral cancer that they have, changing that genetic background, changing the genetics behind it, and expanding it and fixing it up, that will further help them survive. That's a good idea too, is to be like, all right, not only can we reintroduce an extinct species, but maybe we can learn lessons of how to keep things that are alive today still alive. This reference genome that we're building. And again, there's variation between individuals, but figuring out the organisms is like base genome to plot up against. They have 99.9% done.
It's actually
for sure there's no genetic diversity here, absolutely, but having something that they can plot against and have a reference genome is critical, because then, so the other update to this story too is they've been looking at a close relative that's also marsupial, and in that marsupial there's a lot of individuals. They've sequenced a bunch of them, so they looked at this other close relative relative and sequenced a bunch of those, and they found that there were 300 sites in the genome that varied a fair bit. And so they're using that as a proxy of where to introduce genetic variants. Is that the right move? No idea, but at least it's a worthwhile approach. Only animals we drove to extinction, other animals that went extinct by natural selection just proved to be not. But Andrea, what if they went extinct because of stuff that we did to the planet? So we didn't directly hunt them to extinction, let's say, but we're the reason why they're no longer around. Is that what then? What then, Andrea? I don't know, some kind of fish, I'm sure, or a bug, that because we chopped down some of the rainforest and they died. Andrea, if we didn't hunt them to extinction, instead we just we changed the climate and then they died because of that. It's a lot of squeeziness. They went extinct due to human influenced climate change. When they still die off if we couldn't provide them with a proper environment? Like if we brought them back, yeah, Liren. Unless we changed their genome in a way to allow them to survive the new environment. But then is that even what you're bringing back?
All these people.
But let's talk a little bit about the biology of the animal as well. So just like I think there's just a cool factor of something like this evolving and existing. So let's delve a little bit into that. Bring them back before the close surrogate goes extinct too, which is also under threat. Yeah. There's no. It's very, very complicated, Jay, and who knows what's gonna actually what the correct answer is. If there's a correct answer here, there probably isn't, right? At the end of the day. Would that count as a new species if we might? I don't know. Liren, I say I don't know because even in our new species of the week papers that we do every week, the definition of what is a species and the threshold for that is different every week that we look at. And so that makes it far harder to have said definition, right? Is it just mammals who pass down the mitochondrial DNA from the mother? No, insects do as well. At least insects do it, in which case I would assume it's more of a widespread thing on the tree. I'd have to fact check on everything else in the tree, but then I'm pretty sure that's the case then, Smikes, is that it's a widespread one. Because in the embryos of fruit flies, they're loaded up with the mitochondria and the nurse cells. Again, all the sperm is contributing as the second set of genetic information. Well, it's not clear, Smikes, that the Tasmanian devil cancer would hop species, but that is an interesting point to bring up as well. Bring them back, but not back into the wild. So Small Feet, you're maybe suggesting bring them back to learn about the technology. And then afterwards, you've gained that technological information and you don't need to use it in order to bring them anything out. Like they're not going to be introduced in the wild. We just have it. The face disease, yes, yeah, exactly, Optic, that is one thing that these researchers are trying to do in bringing back the thylacine. They're trying to use that component of the introduction of the genome and genetic diversification to bring back things for the Tasmanian devils as well. And is it our place to help them out, or is it such a natural process? For the cancer spread, Jay, it's because they genetically bottlenecked. There are just fewer animals alive today that are very, very genetically similar, and it facilitated there to be a disease spread. The reason that there's so few, that's because of us, humans. And so the question is, okay, is it then our responsibility to fix that, to make it so that that's no longer the case and we help them? Or is it outside of our control, so it's not our responsibility? Or is it something in between? They are, yep, 100%, Jay. And I don't, like I said, I don't have the answers. There is no answer yet. And that's the beauty. That's like our conversation again with Ultra, of where it's a big exercise in trying to identify these differences. And it's like, well, what can we inform each other and learn? We're learning together, Jay. Unfortunately, Neil, those are far few in between nowadays, just because there's not the money for it, right? If there was the unlimited funds that you could just do cool stuff, it would be probably a bit easier. For, again, just the quick hitting update on this is that they finally sequenced 99.9% of the genome that they think is accurate. Again, it is the most complete genome of any species to date, of something that's went extinct before modern technology and preservation started going on. They have also put in 30 genetic markers, or 30 genetic variations, into the cells of the fat-tailed dunart, which is the surrogate host. That's that little marsupial mouse-looking creature. Their reproductive technologies are still being progressed as well, so the team is developing and optimizing ways to induce ovulation in the mouse-looking marsupial creature. And they even have been able to culture single cell embryos halfway through the pregnancy in an artificial uterus. So this is similar to what the mammoth people are trying to do, but they've actually made it halfway through development in this artificial uterus device. So, and again, they say in a recent press release that not only is this a major milestone for the Thylacine De-Extinction project, but advancements by Colossal, which is the company, in addition to the University of Melbourne, for improving assisted reproductive technologies in marsupials that can be applied through the marsupial phylogenetic tree. So I thought that was a pretty cool update on our thylacine friends and what was going on with them.